Hello, and welcome to the Drug Discovery World podcast. My name is Giles, and I'm here to take you through another topical and insightful article from Drug Discovery World. Today's episode is taken from our spring 2017 issue, and is titled Spheroids, Rapidly Becoming a Preferred 3D Culture Format. The article was written by Dr. John Comley, who is Managing Director of HTS Tech Limited. So now on to the main article, Spheroids, Rapidly Becoming a Preferred 3D Culture Format. There are many advantages associated with culturing cells in three-dimensional 3D versus conventional two-dimensional 2D tissue culture. Scaffold-free 3D culture systems that generate spheroids and other similar multicellular aggregations have proved useful as they offer an easy route to access 3D culture and transition into plate-based higher throughput. The emergence of ultra-low attachment, ULA, or cell-repellent plates that generate spheroids has been a major factor in increasing investigation and adoption of 3D culture. The popularity of ULA plates for spheroid generation has shifted the emphasis in spheroid generation to size and shape consistency, to find co-cultures with primary cells, and optimizing conditions for those difficult-to-grow cells. Much progress has been made in recent years in the supporting literature, culture tools, detection technologies, reagents and service offerings enabling spheroids to be adopted as the preferred in vitro 3D platform in safety testing, disease modeling, and drug discovery applications. Spheroids are also proving invaluable in regenerative medicine and tissue engineering research, where they are used as a biological unit or building block to merge into a more complex tissue or organ structures. Interest in three-dimensional cell culture in drug discovery has intensified in recent years, as the potential advantages of such systems over conventional two-dimensional tissue culture become evident. The benefits include the ability of 3D to provide a more physiologically relevant environment, organ-like microarchitecture, morphology more predictive of disease states, and in vivo-like drug responses. One type of 3D structure generated in vitro using cell culture systems is spheroids. These in their various forms may also be referred to as microtissues, embryoid bodies, organoids, or tumor spheres, and typically are spherical aggregates of proliferating, quiescent, and necrotic cells that in culture retain 3D architecture and tissue-specific functions. In recent years, 3D spheroid culture systems have come to the fore in the areas of cancer research, drug discovery, and toxicology. Some of the reasons why interest in spheroids has taken off include the similarity to the way tumours develop and therefore have relevance to studying tumour biology. They are amenable to generation in large numbers of relatively consistent size and shape, and they can be counted, morphologically measured, and analysed by a variety of techniques and instruments. This has spawned significant vendor innovation in spheroid generating consumables, as well as application notes on how to undertake their image analysis. Some of the initial research on spheroids focused on their formation in hanging drop cultures, although the automation of spheroid creation and handling in hang drops plates has hindered the widespread adoption of this approach. Subsequently, a range of alternative microplate-based methods have emerged which are simpler to initiate, offer low cost of entry, and are amenable to higher throughput and screening. The most successful of these is the ultra-low attachment, ULA, or cell-repellent plate. A variety of chemistries, surface coatings, are used to generate the ultra-low cell attachment, but the effect is basically the same, i.e. cell immobilization is inhibited, cells are forced into a suspended state, and they clump together, enabling 3D spheroid formation. ULA plates have contributed to establishing a momentum behind spheroids that has fostered additional developments and a growing library of supporting and validating publications. In October 2016, HTS Tech undertook a market survey on 3D spheroid culture technology. The main objectives of this survey were to understand respondents' current interest in, experience of, and progress made in applying 3D spheroid culture and generation technologies. In this episode, highlights from the market survey are reported, and the findings are discussed, together with vendor updates on their latest 3D spheroid culture technology. Why have spheroids gained traction? Survey respondents ranked a growing library of supporting and validating publications, as the most important criteria for choosing to focus research on 3D spheroids versus other approaches to 3D culture. This was followed by relevance to studying tumour biology, for example hypoxic core, ease of initiating and undertaking investigations, and wide selection of supporting consumables, assay-ready spheroids, and services. Ranked least important was the momentum established behind spheroids. Main interest and activity involving 3D spheroids. The main area of interest and activity involving 3D spheroids to survey respondents was development of improved assays, cell culture systems, and models at 30%. This was followed by regenerative medicine, tissue engineering at 15%, tumour biology at 15%, stem cell research at 12%, basic research at 9%, primary screening, high throughput screening at 8%, toxicity testing, ADME safety assessment at 5%, other at 3%, and then secondary screening, lead optimization at 3%. Awareness and use of technologies. The approaches technologies survey respondents had greatest awareness of today, i.e. most heard about and or most used, to generate or culture 3D spheroids, was hanging drop plates with 92% aware. This was closely followed by ultra-low attachment ULA plates, coated with hydrophilic hydrogel at 90%, surface coatings that mimic the basement membrane and extracellular matrix at 86%, self-made hydrogel 3D forms in dish culture at 83%, and then ULA plates coated with biocompatible synthetic polymer at 79%, and respondents finally were least aware of microspace plates with non-adherent coating at 54%. The commercial technologies or products for 3D cell culture, generation, assay, most used by survey respondents, were Corning, Costar, ULA hydrophilic hydrogel coated plates with 41% using, Greiner Bio 1 CellStar cell-repellent surface plates at 14%, and Insphero's Gravity Plus hanging drop system or Insphero's Gravity Trap ULA plate with tapered ledge, both at 12%. Least used for 3D cell culture, generation or assay was NOF America AMS Bio Lipidur coat plates with biocompatible synthetic polymer film with no respondents using. Preferred 3D spheroid culture format. The kind of 3D spheroid culture format survey respondents most want to achieve used a round, U-bottomed 96 well microplate, had a single spheroid per well or drop, and the spheroids were 100 to 200 UM diameter, with a significant proportion, 42%, of survey respondents indicating they have a specific requirement to transfer spheroids out of their original culture vessel. The type of spheroid culture survey respondents are most interested in investigating was both proliferative and quiescent spheroid culture at 56%. This was followed by proliferative spheroid culture with media exchange, not metabolically stressed, high glucose at 37%, and then quiescent spheroid culture, no media exchange, nutrient deprived, low glucose at 6%. The cell types most used by survey respondents to generate 3D spheroids today were tumour cell lines at 38% of all spheroids. This was followed by primary cells at 19%, and then iPSC-derived cells at 14%. All other cell types had 10% or less use. Survey respondents reported a median of 2 different cell types per spheroid generated today. Expectations for primary screening. Survey respondents' expectations from primary screening using 3D spheroids versus conventional 2D tissue culture was more predictive screening results at 67%. This was followed by different screening results at 21%, NA or do not know at 10%, and comparable screening results at 2%. Assay types and detection instruments used to assess or monitor spheroid cultures. The assay types survey respondents have most applied or plan to apply to isolated spheroids was cell viability, for example luciferase or ATP assay, at 75%. This was followed by cell proliferation assay, for example GFP fluorescence, at 63%, cell differentiation at 55%, and then high content screening or live cell imaging at 47%. Least interest in applying was electrical impedance spectroscopy and acoustic photoacoustic imaging. The microscope phase, fluorescence, or confocal, was the detection instrument most frequently used by survey respondents to assess or monitor spheroid cultures. This was followed by the multi-mode plate reader and then PCR cycler. Least used was the NGS sequencer and a specific spheroid imaging device. Survey respondents rated cell viability assessment as what they most wanted to achieve from monitoring or assessing spheroid cultures. This was followed by size measurements, perimeter, diameter, estimated volume, sphericity, etc., cell proliferation, and then detailed histology and cellular morphology. All respondents least want to achieve protein interaction, FRET, BRET, from assessing spheroid cultures. Leading players in spheroid generation and culture. The vendor or supplier that first comes to mind as the leading player in spheroid generation or culture was Corning, at 38% of all vendor selections. This was followed by Insphero at 22%, 3D Biomatrix at 17%, Thermo Fisher Scientific at 12%, Curaray at 2%, Griner Bio1 at 2%, Sigma Aldrich at 5%, and all others at 5%. It should be stated here that this is not a market share projection, but an indication of brand awareness. Latest developments in 3D spheroid culture technology. The following vendor snapshots describe some of the latest developments in 3D spheroid culture technology. By using thin polymer films integrated into standard or custom formats, 300 microns, www.300microns.com, has developed a new 3D cell culture platform, microcavity arrays. Typically 300 micrometers in diameter and up to 300 micrometers deep, the arrays can be used for static as well as dynamic 3D cell cultures. The microcavities can be manufactured with pores ranging from a few nanometers to several micrometers, so that popular assays, such as transwell assays or assays with active medium flow, can be realized. One of the standard formats, the 96-well microtiter plate, is able to generate 16.224 96 by 169 3D aggregates in a single experiment. Because the design of the array is self-referencing and repetitive, the position of each of the microcavities, and thereby the 3D aggregate, is known and fixed, making it very easy to find and retrieve single aggregates during long-term experiments. The production process allows many materials to be processed, such as tissue culture polystyrene, polycarbonate, cycloolefin polymer, cycloolefin copolymer, and fluorinated ethylene propylene, etc. Moreover, the polymer surface can be modified so that the platform can be used for easy and rapid spheroid generation without the need of an aggregate transfer. Microcavity arrays have successfully been used for kinase profiling, embryonic and adult stem cell differentiation, toxicity assays, and more. Therefore, it is believed the MTP-based microcavity array platform is an ideal high-throughput, high-content screening assay tool. Agilent Technologies, www.agilent.com, has developed a 96-well spheroid microplate, which enables measurement of cellular energy metabolism in single spheroids using the Seahorse XF analyzer. This system provides a metabolic signature based on a set of fluorescent sensors that measure oxygen consumption and proton production within a small transient microchamber. Metabolism is a crucial component of disease pathology and is highly sensitive to microenvironmental factors, phenotype, and cell morphology. These signatures provide valuable insight into the pathology of disease and can be used to elucidate a better understanding of drug interaction, toxicology, immune response, and phenotype changes due to differentiation and mutation. To date, several studies have shown significant differences in response to drug interaction between 2D and 3D models. For example, A549 cells prepared as both 2D cultures and 3D spheroids show significant differences in response to an anti-cancer chemotherapy drug. These experiments were carried out in a single assay plate with multiple time points of exposure and drug concentration. In other experiments, hepatocellular carcinoma cell line, HepG2 spheroids, were used to access mitochondrial impairment in response to drug-induced liver injury, DILI. The workflow for the Seahorse assay is highly flexible and conducive to running experiments with multiple tissue types, co-cultures, organoids, and tissues. The Cultrex range from AMS Bio, www.amsbio.com, is based around a set of extracellular matrices optimized for different applications, allowing the researcher to direct cell activities by controlling their microenvironments. Cultrex spheroid invasion and proliferation viability assays use a dilute, non-gelled, ECM solution to stimulate the formation of cohesive spheroids in low adhesion environments. Such spheroids can be embedded into a gelled invasion matrix to model cell invasion from the tumour spheroid. Human mesenchymal stem cells, HMSCs, and vascular cells can be added to model the role of stroma and vascular tissue respectively in a 3D triculture model, producing microtumours that exhibit tumour morphology and drug response similar to published xenograft data, thus demonstrating a more physiologically predictive in vitro model. The reduced growth factor format of Cultrex basement membrane extract 2, BME2, extracellular matrix has been shown to work well for growing organoids, especially using techniques based on the LGR5 stem cell marker WNT signaling system, pioneered by Hans Clevers and co-workers. More recently, an additional formulation of Cultrex BME has been developed, known as BME R1. This matrix provides a proprietary formulation that has higher tensile strength when compared to other Cultrex matrices, BME1, BME2, and BME3. BME R1 has a higher concentration of intactin, one of the BME components that connects laminins and collagens, reinforcing the hydrogel structure. BME R1 is recommended for use with difficult-to-grow organoid cultures. Researchers have been using Corning, www.corning.com/lifesciences, Matrigel matrix, and Transwell permeable supports for more than 25 years to mimic the 3D surface that cells naturally grow on. Building on this pioneering experience, Corning has introduced new surfaces and tools for manual and high-throughput 3D cell culture, including 96-well and 384-well spheroid microplates and HTS Transwell tissue culture systems. Corning spheroid microplates have round, well-bottomed geometry and are coated with ultra-low attachment surface, resulting in the formation of highly reproducible, singular multicellular tumour spheroids centred in each well. Corning has recently demonstrated a 3D model to study cancer immune cell interactions by combining its 96-well spheroid microplates and HTS Transwell 96 tissue culture system. Corning HTS Transwell 96 permeable support systems are commonly used for drug transport and migration invasion studies. By replacing the standard 2D flat-bottom Transwell receiver plate with a Corning spheroid microplate, the ability to investigate items such as immune cell homing, tumour cytotoxicity, and tumour immune evasion in an easy-to-use 3D high-throughput assay is possible. As more labs transition from 2D to 3D cell culture, the company also provides expert technical support for its customers who are either just starting out or exploring more advanced techniques such as 3D HTS. Greiner Bio1, www.gbo.com/bioscience, has expanded its CellStar line with a cell-repellent surface to address the specific needs in 3D cell culturing. Vessels with a cell-repellent surface prevent effectively cell adherence and offer a straightforward, easy-to-handle solution for 3D cell culture. The cell-repellent product portfolio comprises microplates ranging from 6 up to 384 wells, various dishes, and cell culture flasks. Round-bottom microplates drive the formation of a single spheroid per well, whereas cultivation in flat-bottom plates results in the formation of multiple spheroids. In addition, microplates with cell-repellent surfaces are an ideal platform for gel-based 3D cultures, as they prevent the formation of adherent 2D subcultures. The cell-repellent effect is achieved through an innovative chemical surface modification. In applications such as microscopic analysis of single spheroids with high magnification, where perfect optical properties are requested, Greiner Bio1 collaborates with Nano 3D Biosciences, N3D, to offer an additional technology for 3D cell culture, magnetic levitation and magnetic bioprinting. The N3D core technology is based on magnetic nanoparticles coated with PDL to magnetize cells by attaching to the cell membrane. The application of weak magnetic forces induces the aggregation of the magnetized cells, either by levitation or printing, to form structurally and biologically representative 3D models in vitro. Magnetized spheroids are kept in place, while liquids are added or removed with almost no loss of spheroids. Various studies have demonstrated that the magnetic particles do not show any effect on the growth characteristics and physiology of cells. Insphero, www.insphero.com, specializes in 3D cell culture technologies for toxicology, diabetes, and cancer biology. The only global supplier of assay-ready microtissues and 3D cell culture-based screening services, Insphero leverages its patented hanging drop and ultra-low attachment plates and proprietary 3D select processes for development and industry-scale production of standardized scaffold-free 3D Insight microtissues. The company offers a broad range of fit-for-purpose liver, pancreatic islet, and tumor models certified for in vitro safety testing, disease modeling, and drug discovery applications. Insphero human liver microtissues are co-cultures of pooled primary human hepatocytes and Kupffer cells, and provide a scalable, highly sensitive, and specific model for drug-induced liver injury (DILI) testing. Likewise, human islet microtissues provide a more reproducible and convenient in vitro diabetes model by eliminating size and heterogeneity and laborious hand-picking associated with using native human islets. Inspheri's process normalizes the endocrine cell composition, providing long-term, robust functionality which is pre-qualified and rigorously QC'd with each production lot. Microtissues are delivered in a user-friendly gravity trap plate designed for safe microtissue handling, or contract screening services using 3D Insight microtissues are also available. Insphero continues to drive innovation in the field of 3D cell technologies, and recently launched the Gravity Trap 384 microtissue platform, optimized for 3D applications using high-content imaging systems. This new high-throughput screening platform features a unique well design to prevent accidental aspiration of microtissues during medium exchange. The L-Plasia 3D Spheroid Generator, www.lplasia.com, is a product line of Cure Co. Limited. The L-Plasia Spheroid Generator's newest product is the L-Plasia Multiple Pore Circular MPC, a device with microholes that is designed to be nested within plates. MPC is 6 devices that fit within one plate. Using this insert design, cell suspension is added across the top of the insert where cells are free to fall into the 650 microholes in the insert device. Spheroids form without further manual manipulation, which can then remain in the plate to undergo differentiation. Upon completion of the desired process, spheroids can easily be harvested from the insert by filling the wells below with appropriate media and touching off the insert to the media. MPC eliminates complications by offering a method to create large numbers of spheroids from the microholes of the insert device. The device format is compatible with manual or automated liquid handling equipment. Also, you can order different microhole sizes. Products complementary to the L-Plasia spheroid generators are available for mass spheroid culture, microtissue engineering, and tissue reconstruction, along with other applications outlined on its website. Millipore Sigma, www.milliporesigma.com, is dedicated to providing researchers with the latest innovations in 3D models and tools. It offers a panel of spheroid-adapted technologies from high-content dedicated systems to low-throughput research-based applications. Corning spheroid microplates are particularly appropriate for HCS spheroid studies, as illustrated in a recent publication where these plates were used to measure the effects of doxorubicin on 12 different cancer cell lines, screened on their ability to maintain a regular spheroid shape in real time. Depending on application and downstream analysis, however, approaches used to generate and study spheroids will be different. For example, it has been shown that the hanging drop system is a well-suited technology for imaging a spheroid's diameter and shape, known to be critical parameters to consider for generating accurate results. Perfecta 3D hanging drop plates have been recently used for this purpose to establish and characterize a method for 3D spheroidal growth of liver cancer cell lines for 5 different cell lines. Technologies also need to adapt to new research challenges. Tumour sphere, spheroids composed of tumour stem cells, culture, is an emerging model for studying and expanding the cancer stem cell CSC population. CSCs and their niche cells are important for tumour progression and metastasis, but specialised reagents are requested to allow CSC proliferation. Millipore Sigma is now offering a new chemically defined serum-free medium that ensures stable tumour sphere formation and proliferation, but also increased ALDH high stem cell population. Organogenics, www.nanocultureplate.com, previously Skyvax Life Science, develops and sells 3D cell culture and organoid culture products with MBL International. The Nanoculture Plate, NCP, one of Organogenics' main products, is engineered with an evenly patterned nanoscale structure on a special plastic film attached to the bottom of the plate or dish. The pattern mimics the extracellular matrix and acts as a scaffold for cells, supports cells to migrate across the plate and form healthy spheroids. The pattern eliminates the need of biological or gel matrix, controls the adhesion of cell to the film, and enhances cell proliferation while cells form spheroids. This gel-free scaffold type 3D platform has some advantages, i.e. high experimental reproducibility, usability for high throughput screening, and high content imaging. There are more than 80 publications referring to NCP so far. Most of them are related to cancer research, with a wide assortment of applications and studies such as signaling, hypoxia, anti-cancer drug sensitivity screen, proteomic analysis, live imaging by using time-lapse imaging apparatus, etc. NCP is also used for toxicology or regenerative medicine research. Organogenics has also developed a new generation NCP called the Takoyaki Plate, which has multiple semi-micro dimples with a nanogrid structure on the bottom. This new plate will provide a stricter 3D cell culture, e.g. control of spheroid size, array of spheroids in the well, and frequent medium change. The nanogrid acts as a scaffold and promotes self-organization. This plate is suitable for organoid culture, drug sensitivity assays, drug permeability assays, and IPS differentiation assays. Culturing cells in 3D enables more natural cellular interactions and can mimic the physiologically relevant in vivo microarchitecture. The use of 3D cell culture systems can therefore reduce downstream costs such as secondary assays, as well as in vivo animal testing, by providing more predictive data earlier. PerkinElmer, www.perkinelmer.com, offers a broad range of solutions for the culture, handling, detection, and analysis of multicellular spheroids. The choice of specialized microplates for 3D cell culture will depend on the cell types used and the readout needs of the assay. Cell carrier spheroid ULA 96 well microplates feature a unique ultra-low attachment ULA coating that enables consistent formation of round spheroids from numerous cellular models. This coating suppresses satellite spheroid formation to enable easier analysis and more reproducible results. PerkinElmer also provides Insphero's 3D Gravity Trap ULA plate for long-term culture and the Gravity Plus Hanging Drop 96 well system for advanced assays that require more consistency or defined co-cultures with primary cells. Such experiments can be automated using a PerkinElmer Zephyr G3 workstation to simplify liquid handling and reduce seeding variability originating from manual pipetting. Spheroid growth can be rapidly quantified by imaging with the N-Sight multi-mode plate reader, which also reads viability endpoint assays such as AT-Plite 3D, or measures secreted proteins from supernatant using alpha-LISA technology. A more detailed understanding of spheroid organization and function, for example bile caniculi formation in liver spheroids, can be achieved after confocal imaging with the Opera Phoenix or Operetta CLS high-content analysis systems, and analysis with Harmony software. There is an unmet need to validate 3D cultures and the performance of assays on 3D spheroids. Most assays available have been designed for use with cells grown as a monolayer in 2D. A major difference between 2D and 3D culture models is the distance an assay reagent must penetrate to access all of the cells. A 2D monolayer of cells grown on plastic is typically less than 10 micrometers thick, whereas a 200 micrometer diameter spheroid presents at least a 10-fold increase in the distance a reagent must penetrate to reach all of the cells. Promega, www.promega.com, has used 3 approaches to adapt assays for use with 3D spheroids: reformulation to improve the ability to lyse cells, modifying the assay protocol, or using a small molecule probe reagent that is able to penetrate 3D structures. To address this problem, they have reformulated the original version of their ATP viability assay to create the Cell Titer Glow 3D cell viability assay designed for use with 3D culture models. The new reagent formulation has improved capacity to lyse cells in large spheroids and a protocol that includes increased physical disruption of 3D spheroids by incorporating more rigorous sample shaking and increased incubation time in the presence of lysis solution. The performance of Cell Titer Glow 3D cell viability assay has been validated with a range of cell types and spheroid sizes by comparison to acid extraction, which is the gold standard method for extracting ATP from tissue samples. For apoptosis detection, to maintain and extract caspase 3-7 activity and achieve lysis of cells in large spheroids, Promega has designed a modified assay protocol that incorporates longer sample shaking and incubation times with its caspase Glow 3-7 assay in the presence of the lytic reagent. Examples where small molecule probes penetrate deep into 3D structures and serve as an alternative to overcome the hurdle of lysing large spheroids include the use of DNA-binding dyes to detect dead cells and probes to detect viable cells, such as the approach used with the real-time Glow MT cell viability assay. To support the fast-growing field of 3D culture, StemCell, www.stemcell.com, offers a portfolio of products for 3D spheroid models of varying complexities. On the simpler end of the 3D spectrum, agro-well plates can be used to generate multicellular spheroids. The microwells in wells design provides an easy way to generate large numbers of highly uniform multicellular spheroids, including embryoid bodies, EBs, for pluripotent stem cell differentiation, and cancer spheroids for tumour modelling or drug discovery research. To culture spheroids generated in agro-well or formed from stem progenitor cells, StemCell provides specialised cell-specific media, including mammocult, mammary and breast cancer cells, neurocult, neural stem and progenitor cells, including brain tumour stem cells, and pneumocult, upper airway, including bronchial, nasal and tracheal cells. To support applications requiring more complex 3D models, StemCell provides specialised culture media and optimised protocols for organoids. Organoids are small 3D clusters of cells that represent mini-organs in structure and function. Intesticult medium can be used to derive and culture organoids from small and large intestinal epithelium. These intestinal organoids, or mini-guts, are physiologically relevant and feature a polarized epithelium that contains all the known cell types of the adult intestinal epithelium. Intesticult medium was commercialized under exclusive license from the Hub Foundation for Organoid Technology. Specialized media to support organoid culture from other tissues are in development, including cerebral organoids, or mini-brains, and gastrointestinal organoids. Driven by the mounting evidence that 3D cell cultures are more physiologically relevant than traditional monolayer cultures, 3D culture systems are emerging as the preferred in vitro platform for disease modeling and drug discovery applications. While multiple scaffold-based 3D culture systems have been around for decades, more researchers have set their eyes on the high-throughput friendly scaffold-free 3D culture systems. The thermoscientific Nuclon Sphera cell culture plate features an extremely easy-to-adapt scaffold-free culture surface that promotes the self-assembly of 3D cell aggregates. The vital combination of the cell-repellent coating and the proper curvature of the well effectively produces a single spheroid in each well with consistent shapes and sizes, which conveniently enables researchers to evaluate cell health and cellular responses in situ by fluorescence and colorimetric-based assays. The development of such 3D culture systems offers particular benefits in cancer biology, where the spheroids share several key histomorphological and functional traits with those of the solid tumours. For example, the evidence of a hypoxic core in the cancer spheroid cultured on Nuclon Sphera plate is demonstrated by leveraging the IMAGE-IT hypoxia probe from Thermo Fisher Scientific. a highly oxygen-sensitive compound that measures hypoxia in live cells using real-time oxygen detector with reversible fluorogenic response. The gradient of oxygen in spheroids, progressing from normoxic cells at the periphery to hypoxic cells at the core, provides an excellent model for assessing novel pharmacological agents and drug delivery methods. Discussion. There seems to be little doubt that cells in 3D form offer more natural cellular interactions and mimic physiologically relevant in vivo-like microarchitecture. There is also increasing evidence that they may elicit a differential response to drugs or stimuli than traditional 2D monolayer cultures, i.e., in 3D they may have different metabolic signatures. Scaffold-free 3D culture systems that generate spheroids and other similar multicellular aggregations have in recent years solicited significant interest from researchers seeking ease of entry into 3D culture and a route to transition into plate-based higher throughput. The emergence of ULA or cell-repellent plates, mainly from Corning and Greiner Bio 1, has been a major contributor in promoting this trend and establishing a momentum behind spheroids that has fostered many additional developments and innovation. ULA and cell-repellent plates are currently the most popular approach to the generation of a single spheroid per well, but other spheroid plate formats may be better suited in creating multiple spheroids, for enhanced size uniformity, for specific cell types, or for niche applications. It is probably fair to conclude that spheroid generation is no longer an obstacle, and the emphasis has shifted more to improving size and shape consistency, defined co-cultures with primary cells, and optimizing conditions for those difficult to grow cells. Some of the new developments reported by vendors in this episode included microcavity arrays, 300 microns, combining ULA plates with other existing microplate compatible products, transwells, to support novel applications in 3D, Corning, redesigning the well shape to prevent accidental aspiration of microtissues during medium exchange, in Sphero and PerkinElmer, a plate insert with microhole hanging drops that facilitates generation and harvesting of large numbers of uniform spheroids, Elplasia, the development of spheroid-optimized assay reagents, Promega Thermo Fisher Scientific, a system for measurement of cellular energy metabolism in single spheroids, Agilent, the use of ECM solution to stimulate the formation of spheroids, AMS Bio, and new specialized culture media for organoids and tumor spheres, Millipore Sigma and Stem Cell. Other vendors report the evolution of their spheroid generating formats to new and improved second generation products, Organogenics and Stem Cell, a sure sign that spheroids are gaining traction. Feedback from the survey reported that just under 50% of respondents have a specific requirement to transfer spheroids out of their original culture vessel, for example microplate well, and that there is still an unmet need for devices or mechanisms to perform this transfer of spheroids without any cell damage or disaggregation that may arise from the manipulation. In conclusion, much progress has been made in recent years in the supporting literature, culture tools, detection technologies, reagents, and service offerings enabling spheroids to be adopted as the preferred in vitro 3D platform in safety testing, disease modelling, and drug discovery applications. It should not be overlooked that spheroids are also proving invaluable in regenerative medicine and tissue engineering research, where they are used as a biological unit or building block to merge into a more complex tissue or organ structures. This article was written by Dr. John Comley. Dr. John Comley is Managing Director of HTS Tech Limited, an independent market research consultancy whose focus is on assisting clients delivering novel enabling platform technologies, liquid handling, laboratory automation, detection instrumentation, assay methodologies, and reagent offerings, to drug discovery and the life sciences. Since its formation in 2002, HTS Tech has published 133 market reports on enabling technologies, and Dr. Comley has authored 60 review articles in drug discovery world. If you've enjoyed this episode and want more from Drug Discovery World, then you can subscribe to the journal and get the latest issues delivered to you via post or digitally, completely free. So just head to ddw-online.com and that's where you can subscribe, as well as read all of our other articles and transcripts. We also have all the original full PDFs of past articles, including this one, in our PDF archive. If you've enjoyed the podcast, then please do leave us a review and subscribe so you get the latest episodes as soon as they're released. You can also follow us on LinkedIn and Twitter for updates and latest articles and news from drug discovery world. Thank you for listening, and we hope to see you in our next episode.